Synthetic microplastics currently flow through your bloodstream. These jagged shards circulate alongside red blood cells, carrying the potential to create physical blockages in the brain. This internal reality contrasts with our long-held assumption that plastic is strictly an external problem, a pollutant that gathers on coastlines and threatens marine wildlife. That perspective was challenged in 2022 when a pioneering study used advanced mass spectrometry to analyze human donors. Researchers officially detected intact polymers, such as the PET used in water bottles, inside the blood of healthy people. The primary biological barrier between the environment and the human body has been breached by synthetic polymers. Recent findings from medical institutions like the Cleveland Clinic are now uncovering the clinical stakes of this internal exposure. Micro and nanoplastics, or MNPs, are being discovered specifically lodged inside atheromatous plaques, the fatty deposits that build up and narrow the interior of human arteries. Statistical analysis shows that patients with these plastic-filled arterial plaques face a significantly higher risk of experiencing heart attacks and strokes. However, epidemiological data has limits. Finding plastic particles near the site of a stroke does not confirm that the plastic was the mechanical cause of the event. To establish a direct causal link, researchers had to zoom in and observe the physical behavior of a plastic particle inside a living blood vessel. The microscopic environment of the blood is a dense, chaotic soup of organic material rather than a clear fluid. Lipids and proteins act as organic camouflage, masking nanoplastics and interfering with the standard tools used in analytical chemistry. If you value learning these types of cutting-edge insights about your own cardiovascular biology, subscribe to the channel. This masking made it impossible to witness the physical trigger of a plastic-induced stroke until a recent imaging breakthrough. Scientists utilized miniature two-photon microscopy, a technology that finally allowed them to look directly into the intact, living brains of awake mice. After introducing fluorescent microplastics into the bloodstream, they tracked the particles as they moved at high speeds through the intricate vascular network. As these particles circulate, they trigger phagocytosis. The body's immune cells recognize the plastic as a hostile invader and attempt to eat it. Because the immune system cannot digest synthetic polymers, the cell mutates. It swells into what researchers call an MPL cell, with the plastic particle remaining permanently trapped and intact inside. This swollen cell now struggles to navigate. Its increased size makes it difficult to pass through the narrow, twisting vessels of the cerebral cortex. This leads to the moment of cerebral thrombosis. The movement comes to a dead stop as the swollen cell becomes physically wedged in the vessel, creating a hard blockade. This physical cellular traffic jam is the mechanical cause of the strokes observed in the clinical data. When a single capillary is blocked, the surrounding brain tissue is deprived of oxygen, leading to localized macro effects. Following these microclots, the mouse models displayed severe neurobehavioral abnormalities, including heightened anxiety and impaired physical balance. This mechanical evidence explains the human clinical reality. This process drives the cognitive decline and fatal strokes seen in global populations. Plastic is now acting as an internal cardiovascular pathogen, creating the physical obstructions that drive clinical disease. There is a haunting paradox in our engineering. The same materials we designed to be indestructible for our convenience are now forming indestructible blockages within our own biology.